Good morning, good afternoon, good evening. This is Mike Jones, Biology 121, Anatomy and Physiology 1, the lecture portion. This lecture is going to start Unit 2. Or make sure you understand that. Unit 1 test does not include this information. The Unit 2 test will include this information. So this starts, as the slide says here, the topics in this slideshow begin Unit 2. Okay, I want to be clear about that. So, here we go. Let me put it on a full screen. I will give you, of course, I will give you these slides you will have available to you. This is 4.6D, so all the other stuff, but prior to 4.6D is on Unit 1. This is Unit 2. It starts with membrane junctions. We talk about the nucleus, the properties of DNA and RNA, how protein synthesis occurs, and the cell cycle. The cell cycle means really what we're going to talk about is what does the cell do when it's not dividing, and when it is dividing, it's called mitosis. We're going to get to that. All right, here we go. Right, so membranes connect to each other. Cells are next to each other, of course, and they have to be attached to each other in order to give them some sense of security or a sense of cohesion. And sometimes they use these connections to talk to each other in cell language. Right, so these kinds of associations take place in three forms. These are the three forms, right? You have what are called tight junctions. The second type is called desmosomes. The third type is called gap junctions. So the first two, tight junctions and desmosomes, do not allow cells to communicate with each other. They just allow a certain amount of adhesion or cohesion of those cells to add structure, to add rigidity, to add a sense of stiffness or form to the cells. They hook together, and that helps them. Each cell helps the other cell next to it and all the cells around it. So a tight junction are proteins that link the cells pretty tightly, as the name would imply. The point here is they prevent substances from passing between cells. I mean, you know, in between the gaps between the cells, stuff can't get through there. So the tight junctions force things to go through the cells themselves rather than in between the cells and find their way, you know, without being. This would be very advantageous in, say, the digestive system. You really want things to be tight so that whatever you absorb into your body has to go through your cells first. So that's a tight junction. Desmosomes, I think a good way to think about this is it's a spot weld. It's a small little area where proteins will bind the cell membrane of one cell to another cell, but they're not as tight as a tight junction. They're more spread out. There are gaps in between spot welds. So they do hold the cells together. They give them shape and substance, but they can allow things to get between the cells into wherever is behind the cells, right? This is one of my favorite words in all of physiology, all of anatomy, hemidesmosomes. Love that word. It's an anchor, as it says, right? An anchor of basal layer cells of the epidermis to the underlying components. So there are some gaps in there. They're just think of spot welds. Think of small protein-containing connections between cells, but there are gaps, and they're not super tight like a tight junction is. But neither one of those connections, the tight junction or the desmosome, does not allow the cell to communicate with the other cell, at least directly, not through that junction. The third one here does allow a cell, and in fact, that's really the point. Here is a gap junction. This is a junction where there is a tunnel, or a cavity, I suppose, a connection between one cell and another cell. As I have highlighted here, it's a direct passageway for substances to travel between cells. Now, it could be ions that travel between cells. It could be an electrical signal that happens sometimes in the nervous system. This is particularly useful in cardiac muscle. Cardiac muscles pass on an electric signal to allow all of the muscle cells of the heart to contract as a unit. They don't all contract all at the same time, but they contract in a pretty short amount of time, and it's because they have gap junctions between the muscle cells that allow a transference of ions that allows the contraction to travel through the whole unit fairly rapidly. You don't see this in normal skeletal muscle. Hey, there we are. Let me get I'm also going to get a little pointer thing. So here's a picture of the three membrane junctions you need to know about, right? A tight junction, there is there's no gap between the cell itself, so there's no space, so anything that wants to enter into this area has to go through the cell itself. It's forced to go through the cell. Can't go in between a cell. On the other hand, this thing called a desmosome, again, is a spot weld. You're going to see far fewer of these in a unit space, and so there can be gaps between those, and those would be in places where it's okay for things to go in between cells. Here's my favorite, right, the hemidesmosome. This would be a skin cell. Now, this doesn't really look like a skin cell, but this would be a skin cell attached to its basement membrane. That's what keeps your skin on your body. So you should be thankful and happy for the hemidesmosome. Gap junctions, on the other hand, have this pore. They allow substances to go between. Usually it's ions. Usually it's a pretty small gap. But it does allow things to go from one cell to another cell. Right, so that's a gap junction. Those are the three membrane junctions you have to know about. All right, next up. The next topic we're going to get into is the nucleus, all the stuff in the nucleus, and there's a lot of stuff in the nucleus. Very, very important stuff, as you already know, I'm sure. So, the structure of a nucleus, it is a separated membranous component from the rest of the cell. Really, the big point of a nucleus, as you probably already know, is DNA is held in there, right? DNA is protected and held inside the nucleus, protected away from the rest of the components of the cell. Around the nucleus, so the nucleus has a double membrane layer, just like the outside of the cell has a double membrane layer. But this double membrane layer also has gaps in it. They're called pores. They're called nuclear pores. So these things are called nuclear pores. Now, there are guards. You have to imagine that there are bodyguards at the nuclear pores that allow some things in and some things out, and other things not in and not out. The one thing that is not allowed, let me emphasize, not allowed to leave a nucleus is DNA. DNA is held inside the nucleus, kept inside the nucleus, and not allowed to go outside the nucleus. The only time you ever find in mammalian cells, that would be you and me, the only time you're going to see DNA outside of a nucleus is when the nucleus itself breaks down just before mitosis occurs. Just before that cell divides into two cells, that's when the nucleus breaks down. That's the only time you're going to find DNA outside of a nucleus because, well, there's no nucleus. So, otherwise, DNA is inside here. It's going to present a little bit of an issue later on when we get to other topics of this lecture. Right. So here's the nucleus. The nucleus includes, of course, this double membrane layer, the DNA inside, and there's another structure that's deep inside the nucleus called a nucleolus. A nucleolus, we'll talk about the function of a nucleolus in a moment, but it stains a little bit darker than the rest of it. So inside the nucleus is a nucleolus and the DNA itself. Now, generally, when you know about DNA, right, you normally see it as chromosomes. These chromosomes are, we're going to talk about that in a moment too. Chromosomes are very, very tightly wound and compacted DNA structures just before mitosis occurs. So that's what you're used to seeing when you say, oh, DNA. I think of this, right, the X, right? Oh, and that's DNA. Well, only at a very specific point in the cell cycle is it like that. The rest of the time, it's sort of free-floating, and it's called chromatin. It's not called chromosomes. Chromosomes are the tightly wound, bound up X-shaped things that you are used to seeing. Most of the time, DNA is in the form of chromatin. So it's this kind of free-floating, very, very thin structure, but free-floating inside the nucleus itself. It's more easily accessible for the reasons we're going to talk about in a few minutes. On the outside of the nucleus, the nuclear membrane, are often ribosomes. We're going to talk about those too. Ribosomes are the protein manufacturing machines. So think about that, or keep that in mind as we get further on down this path of this lecture. All right. Now, in words, the nuclear envelope and the nucleolus, right, as it says, I've already told you, it's a double phospholipid membrane enclosing the nucleus, very similar to the outside of the cell. I mean, it's got some different chemical compositions, but for our purposes, it's the same thing. Right? It separates cytoplasm, which is inside the cell membrane, from the fluid that's inside the nucleus. And in fact, that stuff in there, the fluid that's inside the nucleus, is called nucleoplasm, just like the stuff outside of the nucleus is called cytoplasm. The nuclear envelope is continuous with rough endoplasmic reticulum. We talked about that in Unit 1. Remember, rough ER is ER with a ribosome attached to it, and so it is a protein manufacturing station, if you will. And I said there are nuclear pores, so there are openings. Right? Open passageways formed by proteins. They allow the passage of large molecules in and out of the cells. Only certain large molecules go. And ions and water-soluble molecules can also pass through. So they're bodyguards, right? They can allow some things to go in and out, other things are not allowed. And as I've said, DNA is one of those things that is not allowed to leave. More about the nuclear envelope and the nucleolus in particular. Nucleus is a dark staining spherical body. Right? So it stains, with the particular stain, it stains more darkly than the rest of the nucleus. It's composed of protein and RNA. This thing, here's its job. The nucleolus function is to produce the small and large ribosome subunits. So it turns out that ribosomes come in big pieces and little pieces, and they have to connect together to do their job. And you're going to learn what that job is. They're produced in the nucleolus. Of course, what you're going to find out is protein synthesis occurs outside the nucleus, while the nucleolus is inside the nucleus. So it has to produce these fragments, these ribosomal subunits, and they have to go outside the nucleus into the cytoplasm to do their job. Of course, that means they can get through the nuclear pore. Now, the nucleolus is not present in every cell. Often, in nerve cells, you're actually going to see more than one nucleolus, which I think it's funny because they say it's not present in all cells, but in fact, it is double present in nerve cells. You'll see often 2 or 3 nucleoli in the nucleus of a nerve cell, simply because there's so many proteins that nerve cells are producing. But then, of course, in sperm cells, there's almost nothing in sperm cells anyway. Well, you'll see that when you get to AP2, we're going to talk all about sperm cells, but not in this course. But no nucleolus in sperm cells. All right, so now we got to go back inside the nucleus. I said was DNA. Well, we got to talk about what DNA is. We got to talk about, and that means we have to talk about what nucleic acids are. So DNA is a form of nucleic acid. So is RNA. We're going to talk about that too. But what does DNA stand for? DNA stands for deoxyribonucleic acid. Right? DNA. So we're going to talk about all of the the components of DNA, the chemical components that make up DNA. Of course, DNA is the genetic material. We're going to talk about, well, what is a genetic material? What does that even mean? What does that word mean? The whole point to DNA is that it stores instructions on how to produce proteins. That's really what DNA is. It has the cookbook, if you will. It has the protein cookbook. And it knows how to make all the proteins that are present in any cell. And proteins are the things that do the heavy lifting. They do the work of the cell. Any cell that is doing some work, and all your cells are doing something at any given time, they're all doing it because they have proteins in them that are doing the work. And those proteins are specific to specific cells. And those proteins work for a while, and then they age and they are destroyed, or they don't work very well later on, and so you need to make new proteins. That's what DNA holds that recipe book, how to make all of the proteins in the body, and that's what the code is. What proteins are we talking about? Well, enzymes, of course. Remember, enzymes lower the activation energy for any chemical process in the body. Blood proteins, structural proteins, muscle and connective tissue, antibodies. Boy, we're talking a lot about antibodies with COVID and the vaccine that's coming out. We want to create antibodies against that virus. Well, that's a protein. Where are you going to find the recipe for the protein? You're going to find it in the DNA. And then, of course, cell membrane components. And there are other proteins too, but these are the biggies. All of that, how to make these proteins, is stored in the form of DNA. All right, so now we're going to talk about that. Now we have some words here that pop up. Genetic information, right? So genetic information is the cookbook, right? Just think of it as a cookbook. It's the instructions to tell cells how to construct proteins. All of that is stored in the sequence of DNA. And we're going to talk about what does that mean, a sequence of DNA. We're going to get into that in a moment. A gene is a sequence of DNA, a particular length of the DNA that contains information for making one. Got that? One protein. One protein is made by one gene. So that's what a gene is. Now there are lots of proteins. There are tens of thousands of proteins that can be made, and that information is stored on DNA. So that means there are tens of thousands of genes, but even all of those genes, even all of those sequences of DNA, don't account for all of the sequences of DNA. There are DNA in your cells contains 3 billion base pairs. You're going to find out what a base pair is in a moment. But 3 billion. Well, I just said there's tens of thousands of genes, and there are tens of thousands of proteins, and that takes up a fair chunk of that 3 billion, but not all of it. So some of that DNA does other stuff. Most of it has to do with telling a particular gene to turn on, that means make that protein, or turn off, stop making that protein. We'll get to a little bit of that in a moment. A genome refers to the complete set of genetic information in a cell. So that would mean all of the genetic information that does something to you, not just in terms of protein synthesis, but also with other aspects of maintaining a cell. Some of that stuff is beyond this course. If you take a genetics course, you'll get into all of that stuff. But the genome is the whole thing, all of the base pairs of DNA. Some of them, most of it codes for, or keeps information for making proteins. Some of it keeps information for other things. And then gene expression. Gene expression is what portion of the DNA is active and what proteins is that part of the DNA making. That means those genes are being expressed. If they're being expressed, a protein is being made from that gene. If it is not being expressed, then it's quiet, right? It's off. We say that the gene has been turned off. So genes turn on, genes turn off all the time, constantly. There are constant signals depending on what proteins a cell needs at a particular time, and that depends on the kind of tissue that it is. Is this a cell that's in the kidney? Is it a cell that's in the heart? Cell that's in the brain? There are different proteins that are made, and so different genes are turned on and off in those different cells depending on the proteins that they need to do their jobs. All right, so we got to talk about the structure of DNA. Now, you've heard of DNA as this thing called the double helix. And indeed, it is a double helix. What is a double helix? I always try to get people to imagine a ladder, right? So you have this ladder. If you have a ladder that you would, climb up under the roof of your house, we'll say, right? It's got sides of it, wooden, aluminum, whatever material it's made of, and they support, each side supports a rung across the support structure. Well, DNA is very similar to that. The only difference here is that the rung, which of course in your ladder is a solid structure all the way across, the rung in DNA is composed of two different things, what we call, they're called chemicals called bases, and we're going to get to the specific chemistry of this in a moment. But there's a base on one side and a base on the other side, and they match up. And so the rung of the ladder is composed of two bases, one on one side, one on the other side. They match up. They are supported by the backbone. And in DNA, we call that a backbone. Right, so you see over here, this is the backbone. There's the backbone on one side. Here's the backbone on the other side. Here's a base on one side. Here's a base on the other side. That constitutes a rung. Now we get down here, so they're showing you the chemistry here. If you get down a little lower, they just sort of shorten things out, right? They show you here's a rung, and the two bases on either side of the rung. And then they keep on going, and they show you how that ladder, now you're imagining a straight ladder with straight rungs on it, right? Now we take your ladder and we're going to twist it. If we twist that ladder, now we get one of the support structures on one side wrapping around the support structure on the other side, and that is referred to as a double helix. Right, so that's what we mean by double helix. We're going to talk about all of these specifics. In fact, I open with this slide in terms of the structure of DNA. Then I'm going to come back to this very same slide after I describe all of these specific points. We'll come back to this slide. So I'm going to leave this slide for a moment, but we'll come back to it. One other thing I want to point out to you is this is how you think of, most of the time, how you think of DNA. You think of it as a chromosome, and this is the chromosome that you have, right? It looks like a big X. It's not really an X, but okay, we'll go with that. Right? You think, oh, that's DNA, that's my chromosomes. Yes, got it. Well, it's rarely like this, only during cell division, cell mitosis is it really like this. The rest of the time, it's more like this kind of stuff, like kind of more loosely bound together. All right. Now we got to back up. We got to talk about what's the structure of DNA. When we talk about structure, we're going to talk about a nucleotide. So DNA is composed of monomers. A monomer is a single unit of the thing we're talking about. So if we're talking about proteins, a protein is composed of an amino acid connected to another amino acid, connected to another amino acid, and another, and another, and another, and another, on and on and on. And they form this, what we call pearls on a string, right? So all of these amino acids are connected together. We call that a protein or a polypeptide, right? That's the monomer of a protein. Likewise, carbohydrates, more complex carbohydrates, are monosaccharides strung, one monosaccharide strung to another and another and another and another, on and on and on. And we call that a polysaccharide. So in this case, we have the monomer of DNA. It is called a nucleotide. A nucleotide has, in terms of DNA, is very consistent with this phosphate group, is the same for all monomers of DNA. The sugar group, the sugar is composed of what's called deoxyribose. So that's the sugar. S stands for sugar. So you have a 5-carbon sugar here. You have the phosphate attached to it. These are the same, always the same for all of DNA. The thing that varies from one nucleotide to the next nucleotide is this thing, the B. The B stands for base. Now we call it a nitrogenous base because it has nitrogen within its chemical complex. The 4 different kinds, and there are only 4 nitrogenous bases in DNA. Right? Adenine, cytosine, guanine, thymine. That's it. A, C, G, T. That's how we shorthand them. That's how you're going to see from now on. You're going to see A, C, T, G, or T. But you have to understand that they stand for adenine, cytosine, guanine, or thymine. And that one of those goes out here on where it says B. So that's called a base, or a nitrogenous base. So when we string these things together, we get long, 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 long chains. And DNA is particularly long. The string of bases can go on for tens of thousands, hundreds of thousands, even millions of bases. Millions strung together, one right after the other. And then, of course, when you have another, that's half a ladder. The other half of the ladder would be bases on the other side of DNA, and they connect up, and then they twist into the spiral that you recognize. Right, so we call the sugar and the phosphate, we call that the backbone. Right, so it says here a sugar-phosphate chain. I'm going to show you a picture of that in a minute. You need to understand this word. There's a word that's coming up here. Complementary. Complementary strands link nitrogenous bases together from one side to the other side. So the rungs, I told you the rungs of the ladder are made of bases, and they are made of bases that are called complementary to each other. They complement each other. They always, write this down, tattoo this on your arm, put it on your forehead, I don't care where you remember this, they always, in DNA, they always pair this way. C with G, A with T. Right? So the way to remember that is cars park in garages, apples fall from trees. Cars in garages, apples on trees. Okay? C, G, A, T, always. So these are called complementary base pairs. You got to get used to that terminology. You're going to see it, you're going to hear it a lot from now on. They are connected to each other or linked together by hydrogen bonds. So remember we told you in Unit 1, I told you covalent bonds are very strong. The sharing of electrons between 2 atoms. That creates a covalent bond. That's the sharing of atoms. The donating of an electron from one molecule to another molecule creates what we call ions. That's another form of bond. That's a fairly strong bonding form. Generally speaking, in biologic systems, you're going to find, or for sure for our purposes, covalent bonds are the strongest. Ionic bonds are the next strongest. The least strong bond is a hydrogen bond. It's a fairly weak bond, but it's strong enough to do the job. I'll show you what I mean. All right, so this is how DNA links together. So we said, so this is a single strand of DNA. You're almost never going to find DNA as a single-stranded entity. It's almost always, I mean, for our purposes here in this course, we can think of DNA as always double-stranded. There are certain exceptions to that rule for certain activities, and I will point those out to you. But at all other times, DNA is double-stranded and in a double helix form. But they show you, just kind of how DNA links together. So you have a phosphate and a sugar. So this is the deoxyribose, and a phosphate and a sugar, and phosphate and a sugar, and phosphate and a sugar, and blah, blah, blah. You can see on and on and on. So we call that the phosphate-sugar backbone. Phosphate-sugar backbone all the way down. So that's what holds the side of the ladder together. So this would be one side of your ladder. And then you have a base, that kind of faces toward the inside. Base, 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 base. So it's got to be one of those 4 bases that we talked about. Now here, over here on this side, the right side of the screen, you've got a double-stranded structure of DNA. Now this is not a misprint, right? It looks like when you look at this side, you say, okay, yes, I got it. Just like my single strand over here. Great, I got it. But over here, it looks like things are like flipped around. That is true. This is called an anti-parallel arrangement. So one side of the ladder is upright, the other side of the ladder is flipped over. I can't quite get my arm to go that way. Now I'm not asking you to remember that it's called an anti-parallel arrangement. I just want you to not be, freaked out or confused when you see this. You don't want to go, wait a minute, that's drawn wrong. No, it's drawn right. And it leads to other things later on when you get into, especially if you take a genetics class, you're really going to get into this anti-parallel arrangement and what it does, what influence it has. But for right now, all we care about is, what we really care about is, here's a base on this side of the ladder, here's a base on the other side of the ladder. Remember I told you cars are in garages and apples are in trees, right? So if I tell you, and I will on a test, I will tell you this. If I tell you that this base is C, cytosine, you automatically know it has to pair, its complementary pairing is with G, right? So cars in garage. So this base must be a G. If I tell you that this base is G, guanine, you have to know the base over there is C, cytosine, because they pair together. Now I know, cars in garages, well, garages hold cars. I mean, it all depends on which way you're going to go. So C with G, G with C. All right, same thing. If this is A, adenine, adenine is here. It pairs. Its complementary pairing is with T, thymine. So A, T. If this were T, this would have to be A. All right? I'm beating a dead horse here, I know, but I want to make sure because it's going to become an important issue when we start to talk about the next kind of nucleic acid, and then how do you make a protein out of that. So you need to start to be comfortable with that idea. So now, I told you I'd come back to that. So they show you up here, right? They show you the hydrogen bonds between this base, nitrogenous base, and this nitrogenous base. And you can see the phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. That's a backbone. Here's the other one, phosphate, sugar, same thing, backbone. So the important part about DNA and holding information, where's that information held? It is held in the sequence of bases. What do I mean by sequence? I'm going to go back for a moment. When I say sequence, I mean this sequence. If I start down here and I say, if this were C, C, G, T, A, T, just making stuff up, right? That is called a sequence. The information that DNA holds is held in that sequence. And we're going to show you how to decode that sequence. That was the trick that was figured out probably 60 years ago. Right? We didn't really know. We could read these things and we could figure out, oh yeah, C, C, G, T, A, T, whatever. Right? We'd say, oh, what the heck does that mean? We don't know what that means. We figured it out. It's a code. Of course, we call it the DNA code. Right? So we come back to this. Hydrogen bonds holding them together. Now the beauty of hydrogen bonds is we have this complementary base pairing that I've told you about now. Apples and trees, cars and garages. They're held together by hydrogen bonds, which are not very strong, but they're strong enough. And it turns out that when you want to pull the one side of the DNA from the other side of the DNA, you can do that. It's not terribly hard to do that because the hydrogen bonds are weak enough to allow that, but they're also strong enough to keep them together into this double helix when you don't want them apart from each other. So that's the beauty of hydrogen bonding. It's not too strong, but it is strong enough. You got to remember this term, complementary base pairing. All right, so here's our double helix. Here's at the molecular level, we can see each different base on one side and the other side, right? And on and on. Now we twist them around each other so we got this nice double helix. And then we twist it tighter, 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 and we have these proteins that are called histone proteins. So it turns out if you took the DNA in one cell, I mean one of your cells, and we stretched it out full length, we had it as a straight line, it would be almost 2 meters long. That's almost 6 feet. Right? That's as tall as taller than me. That's one cell. How the heck do we take 6 feet of anything and smush it small enough and jam it inside a nucleus and still have access to it so that we can make proteins out of that? It's pretty tricky. But one of the ways we do that is we take that double helix, we wrap them around these proteins called histones, and then those histones wrap around each other, and they create a tight enough coil of chromatin inside the cell so that we can do stuff with it. It's at times of cell division, when we're going to get to in mitosis, when cells mitose, they have to coil the DNA up even tighter than normal. Wrap it around, and when they really, really, really coil up the DNA, it turns into this structure that we call a chromosome. Chromosomes are incredibly tightly wound up DNA. They're like really, really tight springs. Okay, so now we're going to talk about that process, DNA replication. So DNA has to replicate itself. We're going to later on in this presentation, we're going to talk about when DNA replicates itself. But for now, we're just going to talk about how DNA replicates itself. And so what you're going to see again is the beauty of this double-stranded, DNA, the beauty of the double helix, is that because of those hydrogen bonds, you can imagine unzipping DNA strands, right? You just pull them apart from each other. Well, if we pull them apart from each other, then we have what we call templates. We have a template of, what used to be half of a strand, and the other half of the strand serves as a template for the other DNA. So this is how we duplicate DNA. We unzip them, and then we just replicate using our complementary base pairs. We can replicate one side of the DNA. We can replicate the other strand of the DNA. We have an exact copy now. So one of those copies goes in one cell. The other copy goes in the other cell. There you go, DNA replication. How does that happen? It's an enzyme. The enzyme that replicates DNA is called DNA polymerase. Okay, so remember, enzymes tend to have this ASE name at the end. ASE. What a polymer, I told you what a polymer is. So a polymerase is going to make a string of nucleotides, and it's going to make them out of DNA nucleotides. So DNA polymerase is the enzyme that will stitch together all of these nucleotides into a continuous structure, and it will be an exact copy of one half of the DNA. And then the same thing will happen to the other side, and you get another copy over there. So we have two copies, each one goes into its own cell. Because that's really the point when cells divide under the idea of mitosis. Mitosis, the point of mitosis is an exact copy. You have one cell, you want to have two cells in the end that are exactly the same as the cell that you started with. So DNA replication, the goal here is to make an exact copy of the DNA without mistakes. 3 billion base pairs, no mistakes. It's a tall order. Now there are other enzymes that connect the sugar phosphate backbone. DNA polymerase doesn't do that part of it. Doesn't matter about those other enzymes. So here's the steps, right? You have to break the hydrogen bonds between the base pairs. The strands unwind and they separate. New nucleotides pair with the exposed bases under the direction of DNA polymerase. And then other enzymes stitch together the new sugar phosphate backbone. Boom. Copy here, off to their separate cells they go. So here it is in picture form. We are going to talk about this whole thing. This is called the cell cycle. Well, what do we mean when we say cell cycle? What we mean is the cell is not always dividing. Sometimes it has to do its work, right? It has to do its job. So, kidney cells have to filter blood, and heart cells have to pump, and brain cells have to, conduct action potentials or whatever. Cells are busy. They got stuff to do. Well, in between the times that they're doing stuff, they need to make more of themselves. So something like skin cells are constantly replicating, because you're constantly going to the shower and you scrub your arm, and all those skin cells, all those dead skin cells, leave because you scraped them off. Well, there are new skin cells underneath there that are constantly pushing up. So they are under a state of constant replication. Other cells hardly ever replicate. Neurons may never replicate, or very, very rarely replicate. So here's our double-stranded DNA, right? It untwists. Here you get a strand over there and a strand over there. And I told you this is the only time that you're really going to find single-stranded DNA. And then new nucleotides come in, complementary nucleotides, and then they're stitched together, and you have a whole copy of that DNA on one side and a copy on the other side. You got new DNA, new double-stranded DNA. Each cell gets a perfect replicated copy. All right, now we're going to talk about this thing that is called, I told you that within the sequence of bases on DNA is a code. That's called the genetic code. I told you it's in the sequence of bases, but we don't know, is it in the sequence of like 1 base or 2 or 3 or 5 or 10? We didn't really know. What is it? What's a genetic code? How do we know? How did we, come to the idea of how many of these nucleotides in a row code for something? Well, it turns out, long story short, if you take genetics, you're going to go through the whole process of how this was discovered. But for our purposes, all we care about is the end. The end of the argument, the end of the story here, is it's called a triplet code. A triplet code means a sequence of 3 nucleotides. So 3 nucleotides in a row codes for a single amino acid in a protein. Now, we're going to talk about how that happens. But just remember, when we say genetic code, what we're talking about is a triplet code. 3 bases in a row, A, T, C, we'll say. Those 3 in a row code for a particular amino acid on a protein. And then the next 3 code for another amino acid on that protein. And the next 3 code for another amino acid, and so on and so forth. So, to make the math easy, if you have a polypeptide, that means a string of amino acids, one next to the next. If you have 100 amino acids all strung together, that's my polypeptide. If I have 100 amino acids, that means I had to have a gene on the DNA that was 300 bases long. Remember, because 3 in a row, 3 bases in a row coded for 1 amino acid, and the next 3 for the next amino acid. So I got 100 amino acids, I have to have 3 times as many bases. It's just simple math. 300 bases gives me 100 amino acids in a sequence. There you go. So the sequence of bases in a gene determines the amino acid sequence in a polypeptide. And whatever those 3 bases in a row, right? 3 to the triplet here, triplet, 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 triplet. Whatever their order is, that's the order of my protein in the end. I can't have, if I want a certain order of my protein, I can't have the triplet codes all mixed up. They have to be in order. So DNA stores the master copy of the genetic code, remains in the nucleus, right? I've already told you that. It's the cookbook. The cookbook never leaves the nucleus. Bodyguards at the nuclear pores say, DNA, you're staying here, you're not going anywhere. The problem here is, We're going to get into protein synthesis. Great, you got the cookbook in the nucleus, okay? But proteins are made, they're actually made out in the cytosol, the liquid portion of the cytoplasm. Right, the liquidy portion outside the nucleus. So you have to get the cookbook instructions out to the cytosol. How do you get it out there? I'm glad you asked. Right. So here's the problem. DNA stores the master copy inside the nucleus. Protein synthesis occurs in the cytoplasm. We gotta connect them. How we connect them? RNA, another nucleic acid. Slightly different. A lot of similarities, but some important differences. RNA is that thing that can bridge the gap. RNA is made in the nucleus and then travels out to the cytosol. And then protein synthesis can occur from that. Let's talk about how that happens. So RNA, one of the differences between DNA and RNA is RNA is often found as a single strand, not double-stranded to itself, a single strand. So that's a big difference between DNA and RNA. Another difference is it's shorter than DNA, right? So remember, I told you that DNA can be, a chromatin, molecular chromatin can be 100,000, a million, literally, or more, bases long, or base pairs long in that case. However, only smaller segments of that sequence of DNA actually codes for a protein. And remember, that part of the DNA that codes for a protein is called a gene. So the smaller segment that codes for a protein is the part that RNA focuses in on. It just says, I don't care about the whole thing. All I want is these 300. We're using my 100 amino acid polypeptide as my example. All I care about, all RNA cares about, is these 300 300 bases in a row. That's all I care about. So you're going to find that RNA tends to be a lot shorter, a lot smaller than DNA. DNA is millions of bases long. RNA may be hundreds or thousands of bases long. So it's shorter. The other huge, big change, big difference here is there is a base missing, or a base replacement, if you will. So RNA has a phosphate. RNA has a sugar. Now the sugar is different. This sugar is called a ribose sugar instead of a deoxyribose sugar, which is where we get the name ribonucleic acid instead of deoxyribonucleic acid. So the sugar is different, number one. One nucleotide is different. You still have adenine in RNA, you still have guanine in RNA, you still have cytosine. That's all the same. The difference here is you have uracil. Uracil is a replacement for thymine in DNA. So uracil takes the place of thymine in RNA. All right? So you have a ribose and a phosphate and a ribose and a phosphate, et cetera, et cetera. You still have that backbone, the sugar phosphate backbone. And you have still sequence of nucleotides. It's important. However, wherever there would have been a T, or where you would have expected there have been to a T on RNA, instead, there is no T in RNA. You replace that with uracil. You still have the same pairings, though, so guanine and cytosine pair with each other. And adenine and uracil pair with each other. All right. So as I said, you still have complementary base pairing, right? A with U. Now, I don't have a tricky way to remember that one. I have apples and trees, but I don't know. Apples and, you have to come up with something yourself, I guess, on that one. So, now, I told you that DNA holds the recipe book. Proteins are made out in the cytoplasm. We got to get the information out there. The process of converting DNA into RNA is called transcription. Transcription is the process of copying a DNA sequence into an RNA sequence. That is called transcription. That happens in the nucleus. The product that you get out of that once you have this string of RNA molecules, that is called messenger RNA, or we all usually refer to as mRNA. Right, that carries the genetic code that it copied from DNA into RNA, and then the RNA leaves the nucleus and goes out into the cytosol. Now, mRNA, you may have heard mRNA in these vaccinations that have come about from Pfizer and Moderna, these companies that have made vaccines against the COVID virus, the SARS-CoV-2. They are making mRNA vaccines. And so what they've done is they've taken a small portion of the spike protein, this spike protein that's around the outside of the virus. That protein was coded for by an RNA inside the virus. Well, we took a small portion of that RNA, they converted it into an mRNA, and they inject it into you, and your cells make a small portion of the spike protein. They don't make the whole virus. The code isn't there for the whole virus. The only thing that is there is for the small portion of the spike protein that we want your immune system to react to. Right? So you're not getting the virus, you're getting a tiny little piece of the virus, and you're getting the piece that we want your immune system to react to. And that is coded for by the mRNA that is injected into your cells. That's what the vaccine is for. It's cool. How do you make RNA? You make it with RNA polymerase, right? You made DNA with DNA polymerase. If you're going to make RNA, you need RNA polymerase. That makes mRNA a copy of DNA, of a section of DNA. Great. Here's the steps in transcription. Okay? You got to have RNA polymerase. RNA polymerase recognizes the correct strand of DNA to copy. Now, the things that we're not going to get into here, but the things that you have to think about are, you have a double-stranded DNA, right? So RNA polymerase comes along and says, oh, yes, I want these 300 bases. So it pulls the DNA apart, right? It breaks through the hydrogen bond. So now you have a strand of DNA here and a strand of DNA over here. How does the RNA know which side to copy from? Don't worry about that. There is a specific way that happens. Not necessary for us to know that. Suffice to say that RNA polymerase does know which strand of DNA to copy from. Thank goodness. Whew. Right, so RNA figures out, oh yeah, I want that strand, not this strand. Great. So it grabs on. It starts to make an RNA copy. Right, it has to unwind that portion of the DNA, has to expose those bases. It makes a complementary mRNA with the appropriate nucleotides that pair up, still complementary to the DNA. Remember, uracil is used in RNA instead of thymine. RNA polymerase knows where to stop. It knows, okay, I got to the end of the 300, I'm done here. There's a special way it knows that. We're not going to get into that. The mRNA is released. The DNA rewinds back into its double helix. And the mRNA now leaves the nucleus through the nuclear pore. So here's a picture of that. Oh, no, wait, not the picture. We'll get to the picture. That comes next. Right, so we got to get through transcription of mRNA. So this is transcription. Here's DNA on the left side of the screen. RNA is on the right side of the screen. Right, here is the genetic code, GCGTA. Okay, G in DNA pairs up with C in RNA. Great. Same as it was in DNA. Okay, GC. Great. Oh, we got a C here. C, G. I got that. C's and G's go together. Great. Oh, another G. Great. That goes with C. Wonderful. T. There is a T in DNA. There's no T in RNA, but that doesn't matter yet because the complementary pairing with thymine is adenine. Well, RNA has adenine. Great. T and A. Aha. The next one's the tricky one, right? If the DNA has an adenine, you would want that to pair with a thymine. But there is no thymine in RNA, so you have to replace thymine with uracil. That's where it gets tricky. So this complementary base pairing still continues on, but wherever you would expect to see a T in RNA, you replace that T with a U, because That's what you have, uracil. Right, so now we have our mRNA over here. That RNA polymerase made, stitched all up. This thing can now leave the nucleus. Now, I'm gonna back up for a moment. I'm gonna describe this, but we're gonna back it up then. Each amino acid is specified by a sequence of 3 bases. I already told you that, right? A sequence of 3 bases on DNA codes for a particular amino acid on the protein or on the polypeptide. Great. That 3-base pair, that triplet code, is called a codon. So you have to think like a spy now, right? That's a codon. So if it's on the DNA as a codon, on mRNA it's also called a codon. So the mRNA transcribed that codon, and now it's held in mRNA code, I guess you could say. But that's still a codon. mRNA goes out into the cytoplasm where protein synthesis occurs. So now we're going to convert that mRNA into a protein. That process, the process of taking the mRNA and converting it into a protein, is called translation. So you got to get your trans words right here. Transcription is DNA to RNA. Translation is RNA to protein. Transcription occurs in the nucleus. Translation occurs in the cytoplasm. So the mRNA leaves, it binds to a ribosome. The ribosome is the protein manufacturing machine. Right? The mRNA is the template. At the ribosome, the genetic code carried by mRNA is used to synthesize a protein. Translation is that process. Convert mRNA to a protein. Right now, hit the wrong button there for a moment. Sorry about that, people. I want to back up. Back up. Thank you. So here is my, this would be, here's 3 bases in a row, right? GCG. That's transferred over here to RNA, CGC. So that is my triplet codon. So this would be called a codon on the DNA. It's still called a codon on mRNA. Great. Forward we go. Codons. There we are again. 3 in a row. Now let's move forward. So here's the process. Here's the whole process. All of it. We start with a double helix of DNA. Great. You want to make a protein from a gene. Here's the gene in this box. We're going to pull apart the DNA along those nucleotides. So we break the hydrogen bonds. RNA polymerase comes along and makes an mRNA copy of that DNA strand. The correct DNA strand. It knows which one. But right, so we make an mRNA copy. Down here, they've kind of blown it up a little bit. So you see the DNA strand over here. TAGCGA, or GAG. Yeah. Right, so T goes with A. Over here on DNA, here's an A. If you were thinking A and T, well, you're on RNA now, so you have to say, oh, there's no T's. I have to replace it with a U. So that is complementary base pairing from DNA to RNA. C with G, C with G, C with G, great. G with C, everybody's cool. A with, U, yep, you gotta have the U on the mRNA, etc. Right, so you made the mRNA. The mRNA leaves the nucleus out one of those pores into the cytoplasm. A ginormous ribosome. Remember I said way back, many slides ago, I told you the ribosome is made of 2 subunits, we call them. One is a big, or we call it a large subunit. The other one is a small subunit. You have to think of these things like clamps. Ribosome comes along, it's looking for mRNA, it finds it, and it clamps. Big unit on top, small unit on the bottom, grabs the mRNA. Now it's going to start to make protein. So here's the next part of the process that you have to now understand. how does a ribosome know what amino acid to put in a string? It knows it because of another kind of RNA that you have to know about. This kind of RNA is called tRNA. tRNA is called transfer RNA. That's what the T stands for. Transfer RNA is this thing up here. That's a tRNA. Here's another tRNA. Here's another tRNA. A tRNA has two ends to it. All right. On one end, there's an amino acid. You can see that. There's a little blobby thing up there. That's an amino acid. So the tRNA is hanging on to an amino acid on one end. Great. The other end of the tRNA has a series of 3 bases in a row, RNA-type bases. This end of it is called an anticodon. So remember, the mRNA had a codon. Each 3 bases was called a codon. All right, so you got 3 and 3 and 3 and 3 and 3. Our tRNA also have 3 complementary anticodons. So these are like, decoder rings from, years past when I was a kid, right? You'd have a decoder ring. Well, that's what a tRNA is. It's a decoder ring. If the right 3 bases in a row matches up to the 3 on the mRNA, it's a match, right? And so it says, aha, I got a match. The ribosome says, great, I will take your amino acid. Thank you very much. And then another tRNA comes along with the match for the next 3 bases. And the ribosome says, aha, great, I'll take your amino acid. Now I'm going to link those 2 amino acids together. So this is what it does, right? Here's the first amino acid. Then the next tRNA it matches these 3 bases. Anticodon matches the codon. Ribosome takes the amino acid. Great. Another tRNA comes along with its anticodon. It matches the next 3 bases on the mRNA. The ribosome takes its amino acid, connects it to the first 2, and so on. On and on and on and on and on and on and on. These tRNAs keep coming along. They keep finding the 3 bases that match them. They take their amino acid and they hand it on over to the ribosome. Ribosome says, thank you very much. And it strings them all together. And so now you get this string of amino acids. Well, that's called a polypeptide. That's our goal. That's what we're trying to figure out. Right? When it gets to the end of the 300 bases in a row, it's made my 100 amino acid polypeptide that's growing and growing and growing. Once it gets to the end of that, it says, oh, well, that's it, I'm done. And the ribosome falls off the mRNA. It's done. And you got your protein. That's it. That's how it works. Now, the mRNA can be read again, and the ribosome can be used again, but it turns out that mRNAs are only good for a little while. What's a little while? Oh, I don't, maybe you can translate an mRNA 10 times, maybe 100 times, maybe 1,000 times. But at some point, the mRNA breaks down and falls apart. So, and that all makes sense, right? You wouldn't want your DNA to make an mRNA and stick it out in the cytoplasm and leave it there forever. You would continuously and forever make that one protein. Well, you don't need that protein forever and ever and ever and ever. You need it at certain times, and then you don't need as much of it at other times. And then you need more of it at another time. That's how the body works, right? To maintain homeostasis. So if you eat something that has a lot of sugar in it, and there's a lot of sugar in your bloodstream, you need insulin. Insulin helps to get the sugar out of your bloodstream into cells. Well, you only need insulin when there's a lot of sugar in your blood. So your pancreas does this. Your pancreas says, oh, I need insulin. I better make it. It makes insulin, Johnny on the spot. Sends out the insulin. Insulin does its work. Insulin falls apart. The sugar in your bloodstream goes down. Everybody's happy, right? Then you eat again. Sugar goes up again. You need more insulin. And so on. So this is what I just said. Protein synthesis. Protein synthesis requires that amino acids are added to a growing polypeptide chain. Polypeptide chain simply means when we add amino acids one to the next, that is called a polypeptide. Transfer RNA does that. It has to have the right 3 anticodon, right? It has to match up. It has to be complementary to the 3 that are on the mRNA. If it is, it says, aha, we match, yay, I've got the right amino acid. It adds it to the chain. And so on and so forth. Ribosome moves down the mRNA. Each tRNA brings its own specific amino acid to the growing protein chain. That's how it works. Right. Now, it turns out that you have 20 different kinds of amino acids. Yes, that's it. All of the proteins that you make in your body are only made of 20 different kinds of amino acids. Now, of course, you can put those together in lots of different orders. I mean, that creates tens of thousands of different kinds of sequences. And so that's why you can make all these different proteins that do different things with only, you might say, well, only 20 amino acids. Yep, that's all you need. It works great. It turns out that with a 3-base sequence, it turns out, if you do the math, we won't do the math here, but if you do the math, it turns out that you have 64 unique possible codons. So these 3 bases in a row can be rearranged in different ways up to 64 times. So it turns out if you have 64 possible codons and each codon codes for a single amino acid, you would say, well, geez, I should have 64 amino acids. Well, you don't have 64 amino acids. So it turns out that some of these codons are, most of the codons code for a similar amino acid or the same amino acid. So different codes can code for the same amino acid. The math just works out that way. Okay. Of course, when you have an mRNA, you say, how does the ribosome know where to grab the mRNA? How does it know where to start? And it knows where to start because of this. AUG. AUG is always, let me repeat, always the start codon. We call it the initiation codon. So, that's what the ribosome is looking for. The big and little subunits, they're scanning the mRNA and they go, aha, AUG. They clamp onto AUG. That's where they know. Now the ribosome says, aha, I know where to start. Then it starts making the protein from there. And it turns out, how does it know where to stop? Well, it turns out there are stop codons as well. So there are 3 different kinds of 3 bases in a row that say, hey, ribosome, you're done. You made it to the end. You can stop now. Right? So there's the initiation codon, AUG, and there are 3 different kinds of stop codons. So it knows, the mRNA knows where to start, where to stop. Right, so here's the whole process. Now we're all in the cytosol at this point. Right? We're out of the nucleus. We've got mRNA here. Here's the mRNA. And this particular picture has said, here are the codons. Here's codon 1, here's codon 2, and 3, and 4, and 5, and each one of them is 3 bases long. And here's AUG, and our ribosome grabs on there. And then it starts, so the tRNA have the anticodon. The anticodon recognizes the sequence. It complements. It's complementary base pairing still. And it has the amino acid that is next. And so you see this thing keeps on moving down. It moves on down. As it moves on down, it adds amino acids to this growing chain. Now they're showing you here, here's codon 1, and here's the amino acid on that codon. And codon number 2, oh, that one specified the amino acid glycine. Oh, and codon number 3 is for serine, and codon 4 for alanine, and so on, etc. On and on and on and on. Until you get to a stop codon. The stop codon says, okay, that's it. We're done. There's your polypeptide. Great. Now, this is the summary, okay? So here's a summary of transcription in the nucleus all the steps. Here's the summary of translation in the cytoplasm, all the steps. That's just what we went through. You can read the slide. It's a nice, compact version of what you need to know. All right, now, all right, so I told you that when DNA replicates itself, it replicates itself not just because it's bored. It replicates itself because the cell is preparing to divide. Right? So you have one cell, you're going to make two cells. You have to double everything. So you need twice as much DNA. All right? That's DNA replication. That happens in a particular portion of this thing that we call the cell cycle. Right? So after a cell divides in two, now you have two cells where you had one. We'll just look at one of those cells. That one cell enters into what we call the G1 phase. G1 is the growth phase, right? That's where the cell is doing whatever its job is, wherever it happens to be. It's going to do the thing that cell is supposed to be doing. Right? We call that the G1 phase. We consider that cell growth. It doesn't necessarily mean that the cell is actually getting bigger and bigger and bigger. Sometimes it is, but most of the time it reaches a certain size, and that's as big as the cell needs to be. But it's doing its cell thing. Right, now, there's a certain point at which the cell has to make a decision. I mean, it's not like it has a brain and it's sitting there going, what do I want to do? But the cell, in order to, it either has, so if it's a skin cell, it is going to almost certainly go right into the next phase. And that means that it is bound for replication. Right? It is bound to go through another cell cycle, another division of the cell to make more cells. At that point, now when you're done with cell division, we'll get to what happens there. When that cell starts to get into G1, we refer to that entire phase, an entire thing from G1 to S to G2, and we'll talk about the rest of these phases. That whole part of a cell's life, interphase, is most of what it does, right? So in a 24-hour, if we were to lay this out on a timeline, 23 hours of a 24-hour day of that cell would be spent in interphase. It's some portion of interphase. It's either doing its thing as a cell, or it is preparing to divide, which means, as I say here, S phase, this is where the genetic material replicates. That's DNA replication occurs here during the S phase, that portion of interphase. And then it prepares to divide, and then it actually goes through the process of mitosis. So mitosis would be the 1 hour out of the 24. 23 hours is spent all the rest of this time. 1 hour is spent in mitosis, actual mitosis. Now, this point here is called the restriction point. At that point, the cell has to decide which way is it going to go. Is it going to divide? Or should it not divide? Or is it out of control? So there are certain internal checks and balances that your cells undergo. And if the cell decides that some of its genes are out of control, it can choose to, and this is going to sound a little creepy, but it can choose to kill itself. That is called apoptosis. Apoptosis is prescribed cellular suicide. Or sometimes you could say it's cellular hara-kiri. But that might happen because if the cell has a certain gene that is making a protein uncontrollably, as would happen in a cancer cell, if the cell recognizes that that gene is out of control, it can choose, or it can decide at that point to destroy itself instead of replicating, which would replicate a cancer cell. And this happens all the time. This happens all the time inside your body, inside mine, inside everybody's. Some cells are deciding not to become cancer cells by pulling the plug on themselves. Now, they all know their internal signals and why they would do such a thing. But that's called apoptosis. So the cell destroys itself. On the other hand, it could remain specialized. It doesn't have to divide. So a nerve cell is a good example. The nerve cell can remain a nerve cell. It can just do its nerve cell thing. It doesn't have to divide. It can keep on being a nerve cell forever and ever. On the other hand, your skin cells really, really want it because you're washing them off and you're, tearing apart your skin. So your skin has to be replaced constantly. So skin cells almost always go through the rest of interphase onto mitosis. They divide, create new skin cells, and they keep on doing that. So it depends on the cell type, and it depends on other things happening inside the cell as to whether it's going to do one of these three things. It will either go through apoptosis and destroy itself. It will not divide and stay a specialized cell and do its specialized cell thing. Or it will divide. That's it. Now interphase, I just told you what interphase is. Right? Most of the cell's life is spent in interphase, at some point in interphase. It spends a little bit of its time in mitosis. So it can do those things. Cytokinesis is right at the end of the last part of mitosis. That is literally where the cell cleaves itself into two cells. That's called cytokinesis. We're going to get into that in just a moment here. Hold on. There we go. Okay, interphase. Very, very active. The cell grows. As I say, it doesn't necessarily have to be getting bigger and bigger and bigger and bigger. I mean, there's a limitation to how big a cell needs to be. It does its normal cell functions. It does, if it's going into S, it's going to replicate its genetic material, and that means it is preparing to divide. Right? Cell synthesizes organelles, membranes, biochemistry, prepares cytokinesis. And so, that's where it's most active. That's where it's going to spend most of its time. Now, here's the breakdown of interphase. Right? We can be in the synthesis phase, where we're actually replicating the DNA. G1, I mentioned to you, was the growth phase, but it's really doing its cell activity. And then G2 is where it's going to, that's the gap between the replication of the DNA and the actual entrance into mitosis. All right, now, mitosis. What's mitosis? The point to mitosis, the absolute point. You're going to, when you get into AP2, you're going to talk later on in the semester about this other process called meiosis. These are both ways that cells can replicate themselves. But the goal of mitosis and the goal of meiosis are very different. Very, very different. The goal of mitosis is to produce two perfect copies of one cell. You have one cell. When I'm done with mitosis, I want two exact copies of that cell. And so on. 2 and 2 and 2. On and on. Perfect, exact copies. That's the goal. There are four phases in mitosis. The four phases, you can abbreviate them this way. PMAT. P-M-A-T. PMAT. Right, so you got to put that in your memory. PMAT. Prophase, metaphase, anaphase, telophase. In that order, always in that order. Prophase, metaphase, anaphase, telophase. Right, so I told you that DNA replicates itself in S phase, and so it's replicated, but it has not condensed. What does that mean? When it condenses, it turns into chromosomes. Right? So that's the form of DNA that you recognize, that we always look at and we say, oh, look, here's DNA. Those are chromosomes. There's only one time in a cell's life that it forms chromosomes, and it is in prophase. Right, and so it condenses down. Centrioles move to the opposite side of the cytoplasm. Centrioles have to replicate. That also happens in S phase. And then it's prophase where the nuclear envelope and the nucleolus start to break down. And I told you the only time that DNA is found outside of the nucleus is during mitosis, when that nuclear membrane breaks down. All right. Next up is metaphase. Metaphase, this is where the chromosomes line up at the middle of the cell. And it's real easy to see, it's real easy to recognize. You'll see all of the chromosomes of a cell all line up right in the middle of the cell. They're ready. They're ready to be pulled to opposite ends of the cell. And that's what happens in anaphase. In anaphase, the chromosomes actually pull apart from one another because they have replicated themselves, right? So you pull one chromosome to this cell and the other chromosome to that cell. Or the replicated chromosome, I should say. One goes here, one goes there. And then telophase, the chromosomes uncondense. They return to a chromatin structure, which is this unwound version. The nuclear envelope reforms. The nucleoli reform. And then the very last phase is cytokinesis. Cytokinesis, where the cell itself actually squeezes itself into two. So we're going to look at pictures of that. Here's the major events. Prophase, metaphase, anaphase, telophase. PMAT. PMAT. Here it is, picture form. Right, so we have a cell here. The centrioles reproduce. Now this is late, they say this is late interphase, right? We haven't actually entered into prophase, because remember, prophase is where the nuclear membrane breaks down. So here's the nuclear membrane breaking down. So now we're in prophase here. The DNA has already duplicated itself. Here it is as chromatin. Here it is condensed up, coiled, tightly coiled into chromosomes. Those things that you recognize. These spindles go to either side of the cell. Now, late prophase and then into metaphase. This is an easy way to identify metaphase. See those chromosomes line up right in the middle. When they line up, you're in metaphase. They line up on the middle of the cell. Now, chromosomes, the duplicated chromosomes, split apart from each other. This is anaphase. I always think of it as, it looks like an A. Now, yes, the A isn't complete. You don't have a little bar in the middle, but I always think they kind of look like capital A's. So now their chromosomes, the replicated chromosomes, are headed to either side of the cell. We head on into telophase. In telophase, the nuclear membrane starts to reform, and the chromosomes now start to relax a little bit. And this is cytokinesis. Cytokinesis is where the cells actually cleave into two separate cells, two exact copies of what you started with. And now, here we are. We're back in interphase again, early interphase. Round and round to go. If it's a skin cell, goes immediately into this and starts doing this all over again. If it's a nerve cell, it'll be a nerve cell forever. It really won't divide. Nerve cells typically don't divide. But look at these actual pictures, and you can see chromosomes line up in the middle. They start pulling apart from each other. That's anaphase. You start to see the cleavage furrow. That's an indication of telophase. And then you're going to get cytokinesis, which is the splitting apart of the actual cytoplasm of these cells to create two cells. And here it is. Here's a picture of that. Here's our regular old cell. We don't know what phase this cell is in, but we look at this electron micrograph here. It's called a false color electron micrograph. We look down here. Aha! We know what phase this is because now we're starting to get that cleavage furrow. Right? We can see that's going to be the dividing line. So we know we're in telophase at this point. And as this contractile ring, so there's a protein called actin. We're going to talk about that when we get to muscle contraction. There's a protein here that will start to squeeze down and pinch off, in a sense, pinch off the two cells. Now I told you the whole point is to have two exact copies. Well, that's the goal. Certainly DNA will be identical, but can have slightly different size and number of organelles. So maybe not all. Maybe the mitochondria, if there were 5 mitochondria in the cell, maybe 3 went to one cell and 2 went to the other cell. Well, it's not going to be perfect exact, but that's the goal. The goal is to have an exact cell copy. All right, cell division has to be controlled, of course. Right? So as it says here, skin cells, intestinal cells, blood-forming cells divide often and continually. Neurons divide a specific number of times, then they stop. They don't reproduce themselves, even if it would be helpful to have it replaced. They often don't replace themselves. This is one of the problems with aging. The tips on chromosomes, what are called telomeres, shorten with each mitotic cycle. So they act like kind of a clock, or kind of a counter in a sense. So as the telomeres shorten, the shorter the telomere is, the more replication cycles that cell has gone through, and the fewer replication cycles it can continue to go through. And that's part of aging as well. You're going to see fluctuating levels of hormones or proteins in cells. As it says, hormones are growth factors for external controls for cell division. And then there's a thing called contact inhibition. Healthy cells that are dividing get to a certain crowded level, and they don't continue. It's the fact that they're contacting each other in a small space that they do not continue to divide. Tumor cells do not follow that principle. They live outside of contact inhibition, so they continually make more of themselves. Well, that's out of control. And so there you go. Tumors can result from a loss of control over frequency of mitosis, so they continue to divide when they shouldn't. Right, so we get into cell aging and cell death. Aging, of course, I like to think it's a normal, continuous process with obvious body signs. Yes, it's true. I didn't always have gray hair, and now I do. Yes, that's a sign of this normal process. That's here I am, still living, but changing. There are smaller and more subtle changes at a molecular level, of course, and of course, what you see externally, it has to be represented in internal biologic molecular changes. So there's reduced metabolic function of normal cells, reduced ability to maintain homeostasis. So it's harder for your body. Your body has to put more effort into maintaining the homeostasis of certain attributes of body function. Normally functioning cells, there typically is a lower number. It depends on the system you're talking about, but generally speaking, some abnormal function of remaining cells, so sometimes these cells escape. I told you about apoptosis. An abnormally functioning cell should destroy itself, but sometimes they avoid destruction, and they continue to live abnormally. We would consider that to be abnormal. They continue to go on, but of course they're going to function suboptimally. There may be a change in the number of organelles or the change in structure of chromatin and chromosomes. There's more aging. I've told you, cell death, there's 2 mechanisms to cell death. One is, as it said, induced to commit suicide. I've already told you that's called apoptosis. That means the cell recognizes it's doing something wrong, it's out of control, and so there's a process by which it can destroy itself. Right? And so here's the molecular mechanism for that. I'm not so interested that you know the molecular mechanism. Just know that it is normal for a cell to sense that it is out of control or doing the wrong thing, and it is supposed to destroy itself. But if it doesn't, problems can ensue. And here's more apoptosis. Of course, there's enzymes that are initiated that help to destroy the cell itself. As I say, the specifics of this are not that important, and not really important to me in terms of testing you on this. I'm just more interested that you know that this is a process that should happen when a cell is not doing what it ought to be doing, I guess. I guess maybe that's the best way to say it. And it is referred to as programmed cell death. So what's interesting about this is during fetal development, there's a stage of development of developing the fingers. A hand develops as a mitten, like Michigan, right? It develops as a mitten with there are distinct bones inside the mitten, but there's no separate fingers. What happens is the cells in between the bones are told to die. And so they are supposed to undergo apoptosis. They're supposed to die. And if they do it all correctly, you should get 5 fingers. But the initiation of a hand is a mitten, like a flipper almost. So apoptosis is the way in which those cells are supposed to die. They're supposed to get out of the way. Now, I mentioned the tumors, right? So this is the last slide. Tumors are uncontrolled cell growth. Tumors live outside of the regular control mechanisms that tell those cells to either stop dividing or stop doing a particular thing that they're doing, right? They crowd each other out, so they grow in heaps and piles. That's called a tumor. So they don't respond to the normal signals that say stop dividing, stop doing what you're doing, kill yourself. Tumor says, nah, I'm not going to do that, which is not good. I know I'm being silly, and certainly this is a very serious topic. I mean, tumors are not funny, but this is where cells simply cannot be reined in by the normal control mechanisms that all other cells have to adhere to. When you get into pathophysiology, we'll talk all about this stuff. But right now, I just wanted to bring it up to you. I believe that's it. Yes. So, how do I get me? Well, I can't get me full screen. Okay, thanks for your attention. Remember, this is on Unit 2. This is the start of Unit 2. And so keep that in mind for future study. Thanks for your attention. Have a great day.